we're producing things called estimated breeding values, or EBVs, starting to do that for hip dysplasia and some diseases in Cavalier King Charles Spaniels, but eventually we'll move on to other breeds and other conditions. We can also use DNA to investigate complex diseases, and we're working on an internet-based mate selection program with the Kennel Club, so that breeders, instead of having to have all this information in your head and try to make sense of all of it, you can actually go online and check out particular matings and work out which is the best one. And we'll talk a little bit more at the end of my talk about that system. But really what I'm going to talk to you about today is this idea of a breeding program or a breeding scheme for long-term health of your breed. And when we're thinking about the general health of a breed, we have to think about it in terms of genetic diversity and managing genetic diversity within the breed. If we have a disease in the breed that we want to get rid of, obviously we're going to select against that disease. And we can do that, but we still have to worry about the long-term health and diversity. And we can use what are called optimized breeding programs, where we can select against a disease, but at the same time we manage the diversity and we try to make sure that no new diseases will pop up in the future and become a new problem in the breed. So as I say, today we're going to focus on this side of the, well, this morning you were talking about the DNA test and how it's developed, but this afternoon we're going to talk about what we do with the information and how we actually will try to help you with making informed decisions moving forward so that the breed stays healthy. So very briefly this afternoon we're going to talk about a gene pool and genetic diversity. What is a gene pool? What is genetic diversity? What's the relationship between genetic diversity and inbreeding? So what is inbreeding? How do we actually manage that? So how do we stop populations losing too much diversity over time? So in the first half of my talk I'll go through some of this terminology and some of these things, what they mean and how we calculate some of the statistics. And then in the second half of the talk we'll focus very specifically on the Mini Bull Terrier. I've analysed the entire Kennel Club database for UK registered Mini Bull Terriers, and we'll have a look at what is the situation, the current situation with respect to diversity and the gene pool. And then we'll talk about how this might impact on future breeding strategies and how the DNA test for lens luxation can be overlaid on what we see from the results of this analysis. So what is a gene pool? So we normally talk about populations and gene pools as a group of individuals that are interbreeding. So it could be Mini Bull Terriers worldwide, because there's some exchange of genetic material between different countries. Or we could focus particularly, for example, on one particular country. And in this case, I've analysed the UK records. So we could say the gene pool that we're looking at is the entire complement of UK registered Mini Bull Terriers. And again, as you heard this morning, all about DNA, and every individual has two chromosomes, two copies of every gene, two copies of every chromosome, one from mum and one from dad. So if we were to look at our gene pool, so here it's a group of Mini Bull Terriers, let's say it's the UK population. Each of these dogs, if we were to look at a particular gene, each dog has two copies of that gene, and they may have different genetic variants or DNA sequences. So if this is the lens luxation gene, then we know that some dogs are clear, they have two copies of the clear allele, so they have two orange circles here, two orange dots representing that. Some dogs are carriers or heterozygous, so these are the dogs with the green allele and the orange allele. And some dogs are affected, so they have two copies of the affected or the disease allele. So two copies of the green gene here, and these dogs are what we call homozygous. They have two copies. So in my slide here, we're just looking at one particular gene in the genome, but of course diversity applies to all 20,000 genes in the genome. If we look at any particular dog, we've got to consider all 20,000 genes, not just an individual gene. And the fact that we see differences in DNA sequence between individuals, this is genetic variation. So it's the amount of differences that we see between individuals. And And we don't want our population to become completely identical at every genetic locus, so completely homozygous, and essentially what we would call it'd either be highly inbred or almost clones. Every individual would have an identical DNA sequence if we lost too much too much genetic diversity. So what we want to preserve is at least some differences between individuals at the DNA level. Every time we make a breeding decision, what we're doing is selecting some individuals from the population. And we're not only selecting individuals, but we're selecting the genes they carry. So we're making a decision about which genes are going to be carried forward into the next generation. So here are 4 selected individuals now appear at the top of the slide. And if we were to breed from them, we generate a new generation of dogs. And because we've selected 4 individuals where the green gene is more frequent, so we've got 2 homozygous individuals in the middle and 2 heterozygous. So compared to my previous slide, when there was perhaps an equal frequency of the orange gene versus the green gene, when we've selected individuals and we breed from them, we're now starting to see that the numbers of copies of the gene green is increasing. So the frequency of that gene is changing as we put selection on it. And as I say, we're not just potentially putting selection on a particular gene that we're interested in, but we're doing that right across the genome. We've picked out 4 genomes here that we're going to breed from, and those genomes, so those 20,000 genes, are being passed on into future generations. So when we make selection decisions, we're not just necessarily selecting genes that we want, but we may be bringing other things along with that, because we're actually selecting a lot of genes in the package, if you like. So we talked about genetic diversity being DNA or level differences between individuals at the level of the DNA. In any population that's small, there's a fixed number of individuals. We're not bringing individuals in from outside the population. Gradually over time we start to lose diversity, because every generation we're selecting individuals and we're reducing the different types of gene that are available to us in the population, different types of DNA variants. And so gradually over time we tend to lose diversity. And another way of looking at this is that what's happening over time is that inbreeding is increasing. Because if you think about it, in a small population, we start off with a small number of founding individuals, and then over time, gradually everybody, all the offspring, become more and more related to each other. So the same thing happens in any population, whether it's dogs, horses, cows, even human populations. So when, for example, villages were very isolated in the old days, there wasn't much travel between villages, then you tended to get people intermarrying within the same village, and inbreeding levels rose over time, because over time everybody becomes related to everybody else. So it's a natural phenomenon, if you like, of what we call a closed or a small population. We can't help that. It's going to happen, but what we can do is manage in animal populations how that happens. So what I thought I ought to do is just run through with you what we mean by inbreeding. So we generally mean it's mating between related individuals. It doesn't matter whether they're closely related or very distantly related. As a geneticist, those are both inbreeding. And what it means is, at the genetic or the DNA level, so in my slide here at the bottom we have a black and white dog who is inbred because his 2 parents are in fact half sibs. They have the same father. And if we look at it at the chromosomal level, so these bars represent the chromosomes. The black dog at the top is the founder individual. He may be carrying a disease gene, or a gene that I've marked as red. That gene has a probability or chance to be passed on to each of his offspring. The probability of a half that each of his offspring will inherit that gene. So in the next generation, what's happened is that in fact both his offspring have inherited that gene. And then when they're mated together in an inbred mating, the inbred offspring has a probability that it will inherit both copies of